picture of a mammalian cell that's actually engulfing E. coli. So these cells of E. coli are being taken in by this mast cell to destroy, right? It's part of the immune system involved here. So when we talk about membranes actually have several functions, right? So they obviously define the boundary of a cell. And as we've briefly talked about, they are permeable, semi-permeable to different molecules and ions and things, right? Okay, so within the membrane, there are a lot of proteins and glycolipids and things like that that have a lot of different functions. So proteins in general are going to play a very important role in the membrane in transporting things and also with cell-to-cell communication, as we'll go into detail today. One of the most important functions of our cell membrane, though, is the electrical properties, right? So we have what we call membrane potential. We'll talk about it. The fact that there are different charges outside the cell within the cell. Okay, so the cell uses this for a lot of different things, but we think of it oftentimes as nerve cells. So nerve cells use this electrical voltage across our membrane to send signals throughout our body. Okay, so when we look at a diagram of a membrane, what is the molecule that makes up the most majority of the membrane? Phospholipids, right? So all these little phospholipids, and embedded in this phospholipid bilayer, we have all these large proteins, and we have these carbohydrates that are attached to some of the phospholipids and some of the proteins, right? Okay, so when we look at the phospholipid structure, you guys need to remember, right, that phospholipid is ambipathic, which means it has a polar region and a non-polar region. So this is very important because it allows it to function as a phospholipid functions, right? It forms these bilayers. So we have this head that's a phosphate head. Is this region polar or non-polar? Polar. Polar, right? And it's these fatty acid tails that are hydrocarbon tails, right? Which are non-polar. So in a solution, if you were just to drop a bunch of phospholipids in water, they naturally form this bilayer. So you can think about it as the fact that these polar heads are attracted to water, right? They're charged, they're attracted to the water, whereas the non-polar tails have no attraction, so they basically get pushed toward the other non-polar tails, and they form this bilayer. So again, this is just looking at the structures of these phospholipids. This is a regular triglyceride, right? Like we talk about fat, which is very similar to a phospholipid, with the phosphate tail being polar and these hydrocarbon tails are non-polar. So there's this little video we'll watch real quick that shows the model of a bilayer, lipid bilayer forming. So these are representing, right, all the phospholipids, and this is all these water molecules. So as the polar regions are being attracted to the water, these tails just kind of get pushed toward each other. And eventually they form a bilayer, which is what happens in our cells in this form. Right, so these are constantly moving. The polar heads are being very active with these water molecules and kind of move around, but they stay facing the water, right, aqueous area, and the tails stay in that hydrophobic region. Okay, so what you end up getting, right, it's not just the layer, it's actually the membrane, cellular membrane around our cell, or we have other membranes right inside of the cell around other organelles. Okay, so another major component in our cell membrane are what we call sterols. So in animal cells, we have cholesterol. I think. Here we go. Okay, so cholesterols are in animal cells, and the phytosterols are in plant cells typically, but they both consist, remember, when we talk about large biological molecules, of these 4 carbon rings, and one end has a hydrophobic tail, and the other is going to have a hydrophilic hydroxyl attached to it. And they might vary, but they have this basic structure. So within this phospholipid bilayer, this cholesterol is embedded in. What region of this cholesterol do you think is up with the polar heads? Right, the hydrophilic end, which is this hydroxyl, right? And the rest of this is kind of embedded in with these hydrophilic tails. Okay, so we have phospholipids, sterols, and then we have proteins as another major component of the, man, I don't know what's going on here. Sorry, you guys. Okay, as another major component of this membrane, right? So proteins that span the full membrane are called integral proteins or transmembrane. And they'll have a region that extends outward out of the cell, it expands through the cell, and then also expands into the cell. These are important, as you can imagine, for different reasons. A lot of it is for cell communication or transport from one side to the other. In the center of these proteins are going to be hydrophobic regions, and then these are going to be, hydrophobic regions and hydrophilic regions on the exterior. So these can be considered amphipathic as well, these transmembrane proteins. There are also peripheral proteins that are found either on either side of membrane. So on the inside of the cell, right up against the membrane. These are not covalently bonded. They usually have some sort of other interaction, whether it's hydrogen bonds or something keeping it bound there, but they are not covalently bonded. And again, they're found either right on the interior side of the membrane or on the exterior. So if we look at a picture, these are peripheral proteins. And, well, it's hard to tell because they're embedded back here, but there might be some on top as well. Well, here's a peripheral one. And then these are transmembrane, right? And you can see all the cholesterol that's in here as well.
So they're also on the top layer of the membrane?
Yes, they can be on either side. So peripheral means on the outside, right? So either outside of that membrane on the interior of the cell or exterior. Okay, so these proteins have a very diverse number of functions depending on their structure, right? So some, as I mentioned, are used for transport. And we're going to talk about this in detail, but it can either be passive, where it doesn't require any energy, or it might be an active type of transport that requires energy from the cell. So remember, in the cell, we get our energy from ATP molecule. So when ATP is in water, it can react to release that N-phosphate group, which releases some energy. And as we're going to continue to talk throughout the semester, oftentimes this phosphate is not just released to the cell, but it's actually attached to something. So here, this phosphate would actually be attached to the protein, conferring that energy to the protein. Okay, a lot of transmembrane proteins are responsible for enzymatic reactions. So our reactions are carried out in different parts of the cell. Some of them are very localized to different membrane regions. This might be the plasma membrane, it might be the mitochondrial membrane, but these membranes that are found in the, these proteins that are found in the membrane oftentimes function as enzymes. We also have signal recognition proteins, right? So if our body releases some sort of hormone as a signal, specific cell types that need to receive that signal will have very specific proteins that have the receptor site to receive the signal. And then it sends the signal into the cell and carries out whatever was supposed to happen from that signal. Okay, a big part of protein function is cell-cell recognition. And this comes from proteins that have carbohydrates attached to them, hence the name glycoproteins. When you see glyco, that means there's some sort of carbohydrate attached here. So our cells are able to recognize ourselves by the specific carbohydrates we produce, and we attach to these membrane proteins. lot of times, these proteins help with intercellular joining. They bind one cell membrane to the next, if this is in that area where you want tightly compacted cells. And they attach to the cytoskeleton to the extracellular matrix. So remember, we have the cytoskeleton just inside that cell membrane that helps give the cell structure, instead of it just being a loose blob, right? That cytoskeleton gives it structure, and then just to the outside, you have the extracellular matrix, which serves for protection, structure, communication. So you'll have these proteins here that connect the extracellular matrix to other proteins within the membrane. And on the inside of the cell, we sometimes even have these peripheral membrane here attached to the cytoskeleton. Okay, so I'm going to talk a little bit more about these glycoproteins and glycolipids. So these are found specifically on the exterior surface of the cell. Again, these are used for cell recognition. So you will never find glycolipids or glycoproteins extending inward. They're always going to be on the exterior surface. And these carbohydrates are what we call oligosaccharides. So what kind of saccharides have we talked about so far this semester? Monosaccharides, polysaccharides, disaccharides, right? So oligosaccharides means a few, right? So they're not these huge polysaccharides like starch or chitin or something. These, but they're longer than disaccharides. So, 5, 20 maybe monosaccharides linked together. They might be branched or not branched. But these are very important for cell-cell recognition communication. And oftentimes, it forms kind of a coat. There's a lot of them on this membrane. It forms kind of a sticky substance called a glycocalyx. So if you have a membrane with a lot of these, and you're looking at stuff, sometimes it's got this kind of coating on it, and it comes from these carbohydrates. Okay, so we just took a test over this. Hopefully you guys can remember where the production of proteins and carbohydrates come from, where they get processed, right, and moved around. So I know we just had that test. Doesn't mean you can forget it, right? So all the material in this course is going to continue to build on itself. So where are lipids manufactured within the cell? Great, the smooth ER. So the membrane that's going to come, that's been formed for the plasma membrane comes from the smooth ER, right? This is all for the endoplasmic, the endomembrane system. And in the ER, it might produce some of the lipids or cholesterol that's formed in this membrane. Then where are proteins formed? The rough ER, right? The ones that are going to be part of these membranes. Okay, so when proteins are produced in the rough ER, if they're just loose inside the cell like this, they'll eventually be secreted usually, right? They might be a peripheral. But if they're going to be a transmembrane protein, they're going to cross this membrane of the ER. So just like this one right here, we have a transmembrane protein. It crosses that vesicle, is released from the ER, right? And then when it joins the Golgi apparatus, it's still crossing that membrane. And in the Golgi apparatus, this is where it might have some modifications, right? Some carbohydrates added to it if it's a glycoprotein or glycolipid. And then when it leaves the Golgi apparatus as a vesicle, it'll fuse with the cell membrane, right? So watch how this kind of works here. We have in this vesicle a transmembrane and some, it looks like peripheral proteins maybe. And as it fuses with membrane, anything on the inside is going to be placed to the exterior part of the cell. So these carbohydrates inside that are either glycolipids or glycoproteins will face the exterior of the cell, right? And then this part of the protein here is on the interior of the cell. So you never get these carbohydrates facing the inside of the cell. There's no purpose for them inside the cell. They're used for recognition between other cells in our body. Does that make sense? Kind of follow that pattern?
Carbs on the outside and protein on the inside.
Well, carbs are on the outside. Proteins can be transmembrane proteins. They can be peripheral, inside or out. So if I asked you where a protein on the exterior surface of the cell would be found inside a vesicle of the Golgi apparatus, where would that be? On the interior or the exterior of the Golgi apparatus? If it's on the exterior of the cell, would it be inside or outside of the Golgi apparatus vesicle? Inside, right? Because watch how these open up. So if you think about, this is a cross section, right? So we're actually looking at a complete little ball of membrane, and it fuses with the cell membrane and opens up, right? So anything on the inside of that vesicle is now to the exterior of the cell. So this is something important to comprehend. It's, if you're having trouble, let's talk about it after class or look at it in your book a little further. Okay, so one example in the medical world of why these glycoproteins, glycolipids are so important is in research for HIV medication. So HIV uses our cell receptors, CD4 and CCR5, to penetrate our cell. So these are glycoproteins, and these little proteins on HIV's membrane binds into here, and this binding helps the virus basically gain penetration through our membrane. Okay, so when they first were trying to create drugs to treat HIV, they were creating drugs that went for CD4 receptor. They actually didn't know about this co-receptor till later on. But unfortunately, the CD4 receptor is really important in other cellular metabolism functions that we carry out. So people taking this drug actually had really bad effects, right? Because the cells weren't able to carry out their normal function. So they started looking at people who carried the HIV virus but were not getting infected with AIDS. And they noticed that these people lacked this co-receptor. So therefore, this HIV virus would kind of connect here, but it wasn't able to fully penetrate into our cells. So then they started making drugs that bound to the co-receptor here, so blocking it, right? So nothing could bind to it. And so then this was not as important for our daily functioning of our cells. So this was able to help a lot of people with HIV not become infected with AIDS. So you don't have to know that whole story, but this is an example, right, of how these glycoproteins, glycolipids are used for cell recognition. So, if I didn't make it clear, HIV was mimicking, right? It had receptors that mimicked our cell receptors and were able to bind to these CD4 and CCR5. Okay, so our cell membranes are called a fluid mosaic model for the reason that the phospholipids are very fluid, they're constantly moving, and it's a mosaic because within this phospholipid bilayer we have a protein scattered throughout, right? Not really a specific pattern. They're found through outer membranes. Depending on the cell type, you'll have different types of proteins. Okay, so if we look at how fluid our membranes are, there's a lot of things that depends on. But we do know that the phospholipids, like I said, move almost constantly. So it's 10 to the 7th times per second that they'll move laterally within that layer, right? About once a month, they'll actually flip to the other layer. So they'll do a crossover, which means that polar head, right, has to move through the hydrophobic region. So it's not as common, but it does happen. Okay, so two things that create different fluidities in our membranes have to do with temperature. So if you think back, remember we talked about triglycerides, about saturated and unsaturated fats being solid or liquid at room temperature, right? But so let's say we have olive oil at room temperature. Is it liquid or solid? Liquid, right? But if I stick it in the refrigerator, what happens to it? It solidifies, right? Same thing if I take butter and I heat it up, what happens to the solid butter? It turns to liquid, right? So temperature affects the fluidity of these membranes in a similar fashion. And it also has to do with whether there's saturated or unsaturated fatty acids attached to the region. So remember, if they're unsaturated, that means they're not saturated with hydrogens because they have a double bond. So unsaturated glycerides have a double bond that gives them a kink, which helps keep them more fluid, right, at warmer temperatures. Okay, so cholesterol is a really important factor that plays into the fluidity of these membranes as well. So at warmer temperatures, is a membrane typically to be more fluid or solid? Fluid, right, at a warmer temperature. So these cholesterol in here act to keep it from being too fluid. So they kind of give it structure. They're not gonna, temperature doesn't affect them. So they give it a little more structure and keep it more solid. Sorry. At cooler temperatures, if you remember, things start to solidify. Those tails kind of start to bind up. The cholesterol, again, are not affected so much by temperature, and they help keep it from becoming too solid. So cholesterol kind of acts as a buffer, if you will, to the fluidity of these membranes. Okay, so it's pretty interesting. If we look at the composition of different organisms in different environments, their membranes have been adapted to whatever their environment is. So in cooler regions, would you think you would have more saturated or unsaturated tails? Probably unsaturated. In colder temperature, you want unsaturated to keep it from being, because you don't want a solid membrane, right? You want kind of a fluid membrane so things can go in and out of your cell. So at cooler temperatures, you probably have more unsaturated, so more kinks to keep it from getting too solid. And at warmer temperatures, so in areas maybe near vents in the ocean, they're gonna have pretty much saturated tails, right, with some cholesterol in there. And one thing that's really neat is organisms that live in environments with extreme temperature changes actually have the ability to change the composition of their membranes as they need it. So if it gets cold at night, they can add in more cholesterol or take that out, change whether it's whether it's saturated or unsaturated phospholipids to help balance those temperature changes. Okay, so do membrane proteins move? You guys know? Some of them do, yes. So the way they figured this out is they took a human cell and dyed all the proteins with one color fluorescence, and then they took a mouse cell and dyed all the mouse proteins with different. So is anyone colorblind in here? Okay, so you probably cannot tell, but these proteins are red and these are green. So I apologize, this is the picture I have, so you'll have to trust me on this. But then what they did was they fused this mouse and human membrane together, and they watched what happened over 40 minutes. And sure enough, the mouse proteins moved to the other side, and some of the human proteins moved to the other side, and it kind of became hodgepodge, right? So these proteins are moving. Some proteins are fixed. They might be bound to that cytoskeleton or extracellular matrix, but a lot of the proteins do move throughout the membrane. Okay, so let's check your comprehension so far. An oligosaccharide, right, so a carbohydrate, covalently linked to a lipid in the lumen, so that's the interior, of the rough ER may end up on the C, right? Extracellular phase. So if it's on the inside of that lumen of the vesicle, when it fuses with the membrane again, it's going to open up the interior of that vesicle, becomes the exterior of the cell. Remember, you have this bilayer, lipid bilayer that makes one vesicle and fuses. So they just swap sides. Okay, membranes are selectively permeable. This is for those of you who are able to read ahead of time. Which of the following molecules will diffuse most quickly across a lipid bilayer membrane?
C.
E.
D.
Okay, why did you say eat?
Are you ready? It's I have it.
That's okay. So when you look at all of these, E is the correct answer. It's N2. Is N2 polar? No. What about all these other molecules or ions? Are they polar? Yes. So the membrane is selectively permeable. Basically, only non-polar small molecules can move across the membrane without some sort of help. Right? So little hydrocarbons, oxygen, nitrogen, carbon dioxide, they can move across the membrane. But polar molecules, like sugars and ions, cannot be moved across the membrane very easily. Because if you think about it, those polar heads are attracted to them, so they just want to stick. They don't want to allow it through. So how do they transport? We kind of hit on it earlier. How do they move through the membrane?
Proteins.
Lateral.
Proteins, right? So transport proteins are going to move these ions and sugars across the membrane. And it can be either passively, meaning these ions molecules are going to move down the concentration gradient, right? It takes no energy, or active transport, where the cell is moving things against their concentration gradient, and it's going to require some energy in the form of ATP. Okay, so let's look at these in detail. When we talk about passive transport, we're really talking about a process of diffusion. So diffusion is the net movement of ions and molecules from an area of high concentration to low concentration. All right, so if we have a beaker of water with this membrane here that's wide enough to let some of these ions through, or molecules. Ions and molecules have kinetic energy, right? They're moving around. They have inherent kinetic energy. And as they move around, every once in a while, they will pass through this opening in the membrane. And sometimes they will pass back. But because there is a greater number of them on the left side here, more will move to the right than will come back to the left until they reach equilibrium. And you get equal concentrations. So you have equal amounts of the molecule moving through to the right as those moving through to the left. So there's no net movement at this point of equilibrium. So make sure you understand that concept of net movement. It doesn't mean that nothing is moving across the membrane. It just means that it's moving across in both directions at the same rate, right? And this can happen with more than one type of molecule, right? So we have different types of molecules on each side and in different concentrations. They will still move across until both molecules or ions reach equal concentrations on each side of that membrane. This just has to do, right? If you have 10 molecules bouncing around on one side and one over here, you have 1 in 10 chance, or you have 10 chances to 1 chance that it's going to pass through that opening, right? So again, it's just going to naturally move down a concentration gradient from high to low. So that's what this passive transport inside a cell. If there's an opening through one of these proteins, and it's higher concentration outside, it's going to move in, or vice versa, right? It's going to diffuse down a concentration gradient. So osmosis is like diffusion, but we're talking about water molecules specifically moving across, here it is, a selectively permeable membrane, meaning that the membrane will only allow water across, but not the other ion or molecule. So solute, if you will. Okay, so if we have a tube of water here with a membrane right here at the very center that is big enough to allow water molecules through, but not these sugar molecules, very green, water will move from where it has a higher concentration to the lower concentration of water molecules. If you want to think about it in a solute way, it's from the lower solute concentration to the higher. It's trying to equalize the ratio of water to solute. And the reason this happens, remember sugar is a polar molecule. So when water is around these sugar molecules, it's going to form that hydration shell, right? It's going to bond to it. Hydrogen bond to it. So you can think about this once those molecules, those water molecules are bound to those sugar molecules, they're not free to move through this membrane anymore because they're part of that sugar molecule. They're too big to fit through. So technically you have less free water molecules over here than you do here. So by chance you'll have more water being able to bounce through to this right side until it's reached equilibrium. So you can see the water level has dropped over here and has risen over here to equal out the ratio of sugar to water molecules. Okay, so I don't want to confuse anyone, but this only works with things that are dissolved in the water, right? If I poured something that doesn't dissolve in water, then water's not going to move to that side, right? So it's something that's dissolved in there. Okay, so you can imagine our cells really work hard to maintain our water balance, right? We need enough water to carry out our daily functions, but we don't want too much. So when we talk about this, we talk about the solution that it's in, the surrounding solutions, ability to cause a cell to gain or lose water. And this is called its tonicity. So if a solution has the ability to make a cell take in water, well, let me take a step back, sorry. Okay, so tonicity is its ability, right? So an isotonic solution means that the surrounding solution has an equal concentration to that inside the cell. So there's no movement, it's equal, of water between the cell and the solution. So water will go out and water will go in, but it's a net zero. Okay, and a solution that has a higher concentration than that in the cell is called hypertonic. So hyper is more, so the solution has more solutes than inside the cell, so the water will leave the cell, right? So through osmosis, water will move towards the solution that has a higher concentration of solutes. If the solution has less solutes than inside the cell, it's hypotonic. So hypo is under, so less solutes inside the cell, water will actually go from the solution to the cell. Okay, so let's look at a picture. Okay, so let's consider an animal cell first, right? So remember, animal cells have a plasma membrane, no cell wall. So isotonic solution, the solutes are equal outside the cell than in the cell. Water goes in and out at the same rate. This is where our cells are happy. We want to be in isotonic solutions. Okay, if we all of a sudden take one of our cells and drop it in a hypotonic solution, that means there's not very many solutes. It's less than what's inside our cell. By osmosis, water will move from this lower concentration to the higher concentration to equal it out, right, of solutes. So it will cause the cell to burst, and we call this lysing. So the cell is lysed. Okay, if we took that cell, this normal cell, and dropped it in a hypertonic solution, it's going to shrivel up because all the water inside is going to go to the solution that has more solutes than it does inside the cell. So it becomes shriveled. Okay, but what about plant cells? Plant cells have a cell wall, right? So they actually are happiest in hypotonic solutions, so solutions again that have less solutes than inside the cell, because they need that water, constant flow of water into the cell to give it pressure. So they have this cell wall that keeps the membrane from bursting, and it creates a pressure called turgor pressure. So we say this cell is turgid, which is normal. This is what helps a lot of the non-woody plants actually be able to stand upright, because of this turgor pressure. So that water is constantly pressing that cell membrane up against the cell wall. So we stick a plant in an isotonic solution. That means the water is going to go in and out at the same rate. It becomes flaccid. So when your plant starts to wilt because you're not watering enough, it's becoming unhappy. It's at this point. Okay? And then if you completely forget to water it, and it becomes a hypertonic solution that it's in, so there's more solutes outside than in, all the water will leave the cell, and it becomes shriveled, but it also, what happens is the plasma membrane actually pulls away from that cell wall, and we call this plasmolyzed. So sometimes you can look through a microscope at these plasmolyzed cells, and you actually can see the cell membrane that's pulling away from the cell walls. Okay, so passive transport across these cell membranes is using facilitated, what we call facilitated diffusion, right? So nonpolar molecules can diffuse like down their concentration gradient just through the cell membrane, right? But ions and polar molecules have to be facilitated, so they use these transport proteins. Some are what we call channel proteins, and they're just what they sound like, right? It's a channel where one or a couple different types of solutes can just move through from high to low concentration, either in or out of the cell. And one in particular that is common in every cell is what we call aquaporins. So aquaporin is a type of protein like this. It's a channel protein that specifically lets water move in and out of the cell based on the concentration gradient, so through osmosis. So back in that picture we just looked at, water would move in and out of the cell through the aquaporins depending on if it was in a hypotonic, hypertonic, or isotonic stage. Some of these channel proteins will be gated, so there might be a little part of the protein down here that actually stops the channel on either end, and different things within the cell will allow that to open or stay shut. Okay, another type of facilitated diffusion, which is passive transport, this is not requiring any energy, uses carrier proteins. So these have an active site that binds to a very particular solute, and when it binds to it, it causes a conformational change in the protein. So remember, proteins have that tertiary or quaternary structure, right? And they're very, they can change depending on what's bound in them. So they have lots of different amino acids with all those R groups, and when something binds to it, it can cause a change in the shape of that protein. And these specifically open up to the other side of the membrane, and this releases the solute either to the interior or the exterior. So carrier proteins. Okay, so let's look at an example. We have a beaker of water that has a solution of 0.01 molar of everything sucrose, glucose, and fructose. And then we have this membrane mimicking a cell inside here that has a 0.03 molar solution of sucrose and 0.02 molar of glucose. Which one, first of all, has a higher, or what type of tonicity is the solution? Is it hyper, hypo, or isotonic? Okay, first of all, is it higher solution? Is it a higher concentration of solutes or lower concentration solutes relative to the cell?
Lower.
Lower, right? So we add this up, it's 0.03 molar of solutes, and this is what, 0.05 molar of solutes. So if nothing can move across this membrane, which way would water move, in or out? Water's going to move from a lower concentration of solutes to a higher concentration of solutes, or from a high concentration of water molecules to a low concentration of water molecules. So it would move from here to in the cell. 0.03 is lower than inside the cell, so it's going to want to move into the cell to help equalize that ratio of solutes to water. Okay, but what if I tell you sucrose can move across the membrane? So which way would sucrose naturally diffuse? Into or out of the cell? Out, right? It's going to go from high concentration to low concentration. So typically, glucose and fructose, I think, are too big to move in and out of the cell. Or I'm sorry, glucose and sucrose. Yeah, glucose and fructose are too big. So they typically have to be transported in and out of the cell. Okay, so that was passive, right? Passive transport is using just the movement of ions or molecules down their concentration gradient. No energy required, but transport proteins are necessary, right? So either channel or carrier proteins. Okay, so what if the cell wants to move something in or out? It gonna require energy. Again, ATP supplies energy in the cell by adding a phosphate to that protein, which causes a change, gives it energy. And these are performed by very specific proteins, depending on the ion or solute that they're moving. Okay, so one example is the sodium-potassium ion pump. So you can see this is a transmembrane protein, right? It goes across both sides, and it has 3 active sites where sodium binds to. So sodium, once it has bound to all 3 sites, an ATP molecule will transfer a phosphate group there, giving it some energy, causing it to change shape and open to the exterior of the cell, which causes the sodium to be released. Okay, so when it does this, it's changing shape. You can even see, here's kind of the shape of the sodium active site, and when it's opened over here, it's totally changed. Remember, these proteins are changing shape. So as it does this, now it has 2 active sites for potassium, which can bind to it. Once those are both bound, it causes a conformational change in the protein, which opens back up into the cell, releasing the phosphate. And then it's ready to receive 3 more sodium and go back and forth. So for every pump, it takes an ATP to move 3 sodium out of the cell and 2 potassium into the cell. So this is one type of active transport. Okay, so there's 3 main functions why the cell would want to do this. One is to take in nutrients, right, that it needs. So sucrose many times, right? Also needs to get rid of waste. So sometimes it wants to get rid of something that happens to be a higher concentration within the cell, but it needs to export it out. But maybe one of the most important reasons is to maintain this intercellular concentrations of these different ions. And it uses, like I said, ion pumps. So hydrogen, sodium, potassium, and calcium are very common ions that are used. The cell actively pumps in or out against their gradient. All of these are going to require energy, and the reason that it's pumping these ions across is to maintain a voltage. So there's a difference in charges outside the cell within the cell. Okay, so this is kind of a potential energy having this voltage across membrane. We call this membrane potential. Okay, so all cells are going to maintain this voltage. And like I said, it's electrical potential energy. The cytoplasm is actually more negative than the extracellular solution that it's in. So if you notice, it was pumping 3 sodium ions out and taking 2 in. All of those ions have a +1 charge, so it's getting rid of 1 extra +1 charge for every 2 it takes in, right? And the membrane potential ranges in our cells from -50 to -200 millivolts. Okay, so again, you see it's taking 3 sodium from inside the cell, pumping them out, and only taking 2 back in. So this is a net charge of -1, right, for the interior of the cell. Okay, so any type of pump, really any type of ion pump that contributes to this maintenance of the membrane potential is called an electrogenic pump. So there are some ion pumps that don't really have to do with this, but most of them be considered an electrogenic pump because it's creating electric charge across this membrane. Is everyone following here this electric charge membrane potential? More positive outside than inside the cell? So one of the most common pumps we see, especially in plants, or we'll see it later in cellular respiration, is a proton pump. So this uses energy to move hydrogen ions from one side or inside the membrane to outside of the cell. Again, maintaining this charge for work. So the reason this is important, not only for, like we said, in our nerve cells to conduct signals to each other, but we have a potential energy here, right? If we have all these positive charges out here and all these negative charges, which way is a hydrogen ion naturally going to want to move? Toward a negative charge or toward another positive charge? Toward a negative charge, right? So you have this voltage, you have this potential energy built up because you have 2 different charges in and outside of the cell. Okay, so co-transport is a type of active transport that uses this potential that's created here. So co-transport is when two things are transported through one protein, and they both have to be present, right? So co-2. So an example is a sucrose hydrogen co-transporter. So a sucrose has to bind to this protein and a hydrogen ion for this to be moved across to inside of the cell. So here, this is done because we have this charge. The hydrogen ions are naturally going to want to go to this side. So since there's a binding side, the energy comes from this hydrogen binding here and moving in here. And as it does that, it takes the sucrose into the cell as well. So you can see this works in conjunction with the proton pump, right? The proton pump is using ATP to continually pump the hydrogen back out to keep this membrane potential there. And then the cell, this protein uses that potential energy to move sucrose into the cell. So co-transport protein moves two different molecules into the cell, right? Okay, so not only is it doing this through the electrical difference, right, that there's positive here and negative, but how many hydrogen ions do we have here, and how many do we have inside the cell? 6 and 2, right? So there's also a concentration gradient, right? So hydrogen wants to move down from a high concentration to low, so we have dual action here. We call this electrochemical gradient. So these hydrogen ions are going to move down an electrochemical gradient because there's a charge difference, right? This works with ions only. So there's a charge, it's going to move electrically down, and it's also going to move down a chemical gradient, right? Because it's a chemical. So from concentration gradient, high to low. Electrochemical gradient are those two forces combined. Does that make sense? I'll take that as a yes. Okay, so what do athletes and people with diarrhea have in common? Dehydration, right? So as we sweat, if you're an athlete, you will not only sweat out water, right? You sweat out lots of salts and electrolytes. So it's important, right? Not only take in water, but replenish those salts and sugars. Okay, so people who suffer from diarrhea get this from the rotavirus. So I'm not really talking about here so much. It's not so much of a problem here, right? But there are thousands, really, I think millions of people die every year from diarrhea across the world in third world countries that don't have sanitary water systems set up. So what happens is people with this condition, the rotavirus just gets shed into their water, and then you don't have clean water. So it's constantly a problem. And if these kids or who suffer from it most aren't rehydrated properly, then you die, right? There's a real simple solution, right? So there are these oral rehydration therapies, what we call it. We think of Gatorade when we think of being an athlete, but actually Gatorade is not a great one for those who are suffering from the rotavirus. The reason is you want a very specific ratio of salt and glucose with your water to reabsorb properly. So a lot of salt is discharged into your intestines and your stool, and then you reabsorb that salt in your intestines before you excrete that out. But in diarrhea, the movement is too fast, so you're losing that salt. And we know salt, sodium, as we just saw, is a major component of maintaining that membrane potential. To reabsorb salt, you actually have to have glucose because it's a co-transporter in your system that moves salt, sodium, and glucose back into your system. So you need a very specific portion as well, because that glucose, if you drink too much sugar, like in Gatorade, it actually becomes a hypertonic solution, which means water is going to move from your body into that solution. And if you're suffering from diarrhea, it's just all going to come out and you're not absorbing anything. You don't have to know that whole story. I'm just giving you a good example of where we see a co-transporter in our body, and maybe something interesting instead of me just repeating what's out of your book, right? Okay. So now let's look at bulk transport. We've looked at ions and small molecules, but what about large molecules like huge proteins or polysaccharides? This is going to also require energy, but it's not transport proteins that move it across. They are processes called exocytosis and endocytosis. So endocytosis actually has three different types we talked about: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Okay, so let's look at this. So phagocytosis is when we have a bulk material the cell wants to take in. And what happens is the cell membrane extends outward and envelops around it, right? So we kind of form these pseudopodia. It's like an amoeba, right? These extensions outward that circle around this food particle and then envelop it and pull it in. It will then fuse with a lysosome, which will help digest it in the cell and distribute the parts out where it's needed. Pinocytosis is when the cell membrane kind of really just dips back inward, and as it does this, all this extracellular fluid and whatever happens to be in it come into the cell. So phagocytosis is an extension of the cell membrane around something, and pinocytosis is thought of as drinking. It kind of takes an inward sip, if you will, of the extracellular fluid. Okay, then we have receptor-mediated endocytosis. This is when our cell membrane has specific receptors looking to bind to something specifically. Okay, so once these molecules have bound to our receptors, it causes the membrane to fold inward, and sometimes it'll take in some other things with it as well, so fluid and other little ions and things. And typically, this membrane is coated with recognition molecules, so it can be whatever it's trying to bind to. It has a signal to the rest of the cell. So now this vesicle has the correct things it's supposed to bind to. The cell can do appropriately what it's supposed to do with it. So these molecules that bind to these receptors are called ligands, and any molecule that binds to or that has a specific receptor is called a ligand. You'll hear that throughout your career in biology. Ligands are just molecules that bind specifically to some receptor. Okay, then exocytosis, I think we looked at briefly in chapter 6, is when we have a vesicle within the cell. This is like when we want to get rid of waste products often, right? So maybe, or something coming from the the Golgi apparatus, right? It's going to have this vesicle. So this is a bilayer, right, membrane, and it kind of forms with the cell here, and then it opens up and releases whatever's inside the exterior. So this could be hormones, it could be waste, different things that the cell wants to excrete from the cell. Yes?
How is it different from the other one?
From one of them.
The ball?
Well, so when we're talking about the Golgi apparatus, it's really not different. It's just that, so let's go back to what you're talking about.
Pinocytosis.
Oh, pinocytosis? So pinocytosis is taking something into the cell, exocytosis is excreting something. Does that make sense? Pinocytosis is when the cell wants to take something inside.
No, exocytosis, how is that different from the other one?
From what?
From
From the
phagocytosis.
phagocytosis is taking something in as well. I thought you were asking about something else. Okay, so phagocytosis is taking something in. It's taking in like bulk food, right? So the membrane is growing around it. And pinocytosis is kind of just coming inward, but it's taking stuff into the cell. Okay, so this kind of sums up the membrane transport pretty well.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
Okay, so sometimes it helps seeing actual images. Which of the following molecules will diffuse most quickly across the lipid bilayer membrane? B, right? So oxygen is nonpolar, it shares electrons equally with a cell. Cells are taken up by other cells by which of the following? B. What was that? As in dog? Yeah. So if you think about a cell, another cell, like a bacterial cell, like the one I showed you the very first slide, is a rather large, bulky item, right? So it's going to be phagocytosis is how the cell membrane, you'll have to grow around that cell and kind of pull it in. Okay, so make sure that you can define what an amphipathic molecule is. So what's an example of two amphipathic molecules?
Phospholipid.
What was the first one you said? Well, phospholipid, yeah. And what else? Right, so our transmembrane proteins. Yep, you got it. Okay, so what are aquaporins? Right, water channel proteins, good. And then make sure you understand diffusion, osmosis, and all the chemicities, right? How membrane fluidity is influenced by temperature, membrane composition, and distinguish between what a peripheral and integral protein are. All these terminologies we went over. Okay, make sure you know how to transport proteins. Transport proteins facilitate diffusion. How electrogenic pumps create voltage. Okay, so what are two types of electrogenic pumps we look at? Sodium, potassium, proton pump. Good. So I think I heard someone say the sucrose hydrogen, is that an electrogenic pump? No, that's a co-transporter. Good. And then make sure you understand all the exocytosis and endocytosis mechanisms. Okay, so we had a short week. That's okay. We're going to pick up on metabolism. If you have issues with understanding energy, like kinetic potential energy, chemical energy, please make sure you read this chapter before Monday, because I think it'll help you understand the lecture better. Okay, you guys have